Ahmed Samir from Cairo University, Biophysics department. I'm a master's student, and my work now is on some simulations that we are doing to design a drug in order to prevent the infection of influenza virus. Okay, so many of you will ask, what is the relation between this and the approach here, which is quantum simulation and the quantum computing. And I'm trying to find out. I tried to find out what should be the relation, and I found a lot of relationships between both of them. First of all, let me introduce you to my work. My work starts with only one idea, that we have a virus called flu, which infects many people, and it can evolve so quickly, okay, and it can mutate, having something called reassortment, which gave it the chance to evolve all the time and create new versions of it. So it's so dangerous, and we needed to eliminate the reassortment of this virus. So most of the antiviral drugs that we are using now, okay, in order to prevent our flu, and to kill our flu, we use something called hemagglutinin antiviral drug or neuraminidase antiviral drug. Let me introduce you what's the meaning of both of them. I'm sorry, I had a presentation for the flu virus, but I'm not so clever in this Macintosh, okay? So I'm presenting this, and I can illustrate it on the other side. First of all, are you all with me? Okay, first of all, this is the influenza virus. It has two proteins on its outside surface, okay? One of them called hemagglutinin, we refer it H, and one of them called neuraminidase, which is N. So you can hear always H1, N1, H3, N5, you know, this is influenza. H1N1 is a swine flu, okay, that evolved in 2009. So it refers to this glycoproteins on the surface of the influenza. So it uses these proteins in order to enter the cell. This is our cell, actually unlucky cell, okay? And after it enters it, it uses its genetic material, which are 8 segments, okay, composed of something called RNB. After entering the cell, it releases its 8 segments and lets them enter the nucleus of the cell. In order to use the cell to reproduce and to produce new parts for this virus, to reassembly and to and to help it infect more cells. Okay, that after these segments enter the nucleus, okay, more parts of this virus will be produced in this cytoplasm, and then they will be reassembled to form new viruses. This will get out of the cell and infect other cells. Okay, so this happens inside the nucleus by something called transcription, which means is to copy the genetic material, or part of genetic material, on something called messenger RNA that will be sent to the cell cytoplasm in order to be manufactured to form new proteins. Okay, so if we prevented this transcription, there will be no proteins, no new proteins, and there will be no new assemblies of the virus. So we kill the virus. Okay. So you will ask me again, what's the relation? How do you relate this? You know, in Cairo University we use theoretical methodology. Okay, only we are not dealing with this in labs. We are using our computers only. So we use servers. And that's for our fortune that after analyzing the thing, we were using this protein, which is called RNA-dependent RNA polymerase, okay? After analyzing it, we found it composed of 3 domains, okay, of 3 subunits, like, I think we can come back here. 3 subunits like that. This is called the PP2 subunit, protein basic subunit, and this is called PP1, okay? And the most important for me is this one, which is called BA, or acidic protein. This acidic protein contains potential parts, potential sites, that we can use them to kill the transcription, to stop the transcription, as it initiates the transcription. So we are now targeting the sites on this BA, okay, as the C-terminal domain of it. So we used many servers, okay, because this site and the binding between the sites and the part of the cell, okay, it wasn't solved, structure solved by crystallography or even cryo-electron microscope. It wasn't solved because, you know, the protein was unfolded and it's, you know, not ordered. So they cannot solve it using electron cryo microscope, and even X-ray crystallography cannot solve it. So the, I can say that we can try to solve it using NMR, because, you know, it deals with cozy proteins and the proteins that are moving. Okay, so after decision of this parts, and after we saw that we cannot use laboratory techniques to solve it or to deal with it, okay, we decided to go to in silico methods. Okay, the in silico methods means that we will predict the conformation of this part, okay, using techniques in silico, okay, which will be in protein modeling. 3 techniques. First of them, okay, thank you. Okay, first of them called homology. Modeling. Homology modeling can be used only if we have sequence or 3D structures that has similar parts to already solved structures, okay, in the database. So in our case, we didn't have any part that was solved like that because it was disordered, it was not solved, so we cannot use homology modeling. Then threading modeling, which combines the homology modeling and something called ab initio modeling, okay, that it can predict something computationally and continue and complete the structure using a homologous parts, okay? And of course, this wasn't so beneficial for us. The last one was the ab initio method, and we use the server of ITASA to predict the structure. Okay, this method depends on, you know, they are placing every atom, okay, in many different positions, okay, something like quantum computing, okay. You don't know where is the position, you just throw probabilities and let them structure together, okay. That's what's being done in ab initio method in ITASA. So it gives you more than one structure, and you can choose which one you need, which one fits for you. Okay, how can I determine which one is the best for me? Using server called saves. Okay, saves is a validating server. Okay, this validating server uses 7 tools, 7 techniques. One of them is verify 3D. Okay, that after we predict a 3D structure of unsolved protein, we can use the server of saves in order to say this model is valid and it can be used or not. Okay, depending on many techniques. So I'm here trying to explain only one tool, which is verify 3D. Okay, which is a part of saves modeling server, a validation server. Okay. this was a paper published in 19s, I think, and it's called A Method to Identify Protein Sequence that fold into a known 3D structure, okay? If you have a fold, if it was predicted or not, you can throw it in this tool, and it will tell you, is it reliable or not? Is it correct or not? Okay, it uses the following concepts, which is inverse folding problem. What's the inverse folding problem? Most of our work is dealing with sequences that we convert them into 3D structure, okay? What if we did the reverse of that? Okay, we use a 3D structure to obtain the 1D structure, 1D sequence, okay? This will give us a certainty that this 3D structure is intimately related to this 1D structure, okay? And that's what we need. We need to make sure that 1D structure, which is a sequence that we have already, and it's so easy and so valid, if it was related to the 3D structure that we predicted or not. So if we use the inverse folding problem or dilemma, we can reverse the folding and make sure if it will get back to the 1D structure or not, okay? That's the idea behind saves. Okay. So they had an older method called Tertiary template method, which depended on, like what I said, that they are searching for sequences that are compatible with the 3D structure, okay? And then they fix the backbone of each of these sequences, okay? And try to change the places of the side chains to say, will this sequence take the same shape of the 3D structure that we predicted or not, okay? But this method had two cones which were differences in shape and size of residues at equivalent positions were not fully taken into considerations. We were not able to differentiate between different amino acids in these places. You can place any amino acid in this place and it won't say no, okay? This was the first one, and the second one, insertions and deletions, which are important concepts in our field, that if you have an amino acid in certain place that doesn't fit in this place, okay, you can delete it or just insert something instead of it. Any amino acid, any other amino acid that can fit in this place. So it doesn't contain this feature, which is deletion. So they invented verify methodology, okay? Verify methodology relies on something called environmental classification. What's the meaning of that? I'll explain. First of all, they have the first stage, which is the environmental classification, that we start with a known 3D structure that we predicted, okay? And then we determine 3 features of each single environment. What's the meaning of that? That we will take every position, okay? Every position in the 3D structure will have 3 environmental classes, okay? The 3 are, if it was buried in polar environment, if it was buried under atoms, not polar, or if it was exposed, only exposed, no bury, okay? So these were the 3 classifications that we are using, okay? And the third one was the preference of the 2D structure, okay? Does this amino acid prefer beta sheet or alpha helix or otherwise, okay? Random coil or something like that, okay? This is the configuration of proteins in 2D, okay? Secondary structure of protein. So, it's like that. That's a 3D structure, okay? This is a 3D structure, the whole 3D structure, and then we can determine this place is buried and apolar environment. It's buried under atoms, under other atoms. And this place is buried, but under polar conditions, under polar atoms and polar amino acids, even water. And this part is exposed, and only exposed, okay? No atoms above it or under it. So these are the three classifications, and then we determine the secondary structure. Which secondary structure will this amino acid take in environment? Alpha or beta or random coil? Okay. After finishing stage 1, we can say that we converted, and this is so important, we have converted 3D structure into 1D string-like environment, like converting 3D structure into 1D sequence. It's the same, but under the conditions of environmental conditions. Okay, second stage, something called 3D structure profile. What's the meaning of 3D structure profile? It's the most related part to the quantum simulation, okay, where we make tables, okay, like, let me explain it. We have about 20 possible amino acids, and each one of them has certain environmental preferences, okay? There are some amino acids that are polar, some amino acids that are non-polar, okay? Some of them are charged, are not charged, you know, everyone prefers something. So we make a table for each position of the 3D structure, okay? Say we have about 3,000 position in the 3D structure. Each position will have 20 multiplied by 3 possibility. How is that? We have 20 amino acids, okay? So each position will take this part, position, will have 20 possibility, okay? Alanine, glycine, anything, okay? The 20 amino acids. Each amino acid can be alpha or beta or random coil, which is other. Each of them has environmental classification, and under the knowledge of the 3D structure, we know where is the polar part and where is the non-polar part. So we give scores to each amino acid according to that, and also according to its preference of being alpha sheet in alpha sheet or alpha helix or beta sheet or random coil, okay? So it's all about probability. We throw the amino acids, we throw the preferences, and we multiply them together. They can get entangled together, I don't know, but this is the most related part, okay, to the work being done here. Okay, then this is the second stage. Okay, this is the table, like what I said. This is amino acids, A, C, D, for each position, and we multiply it, E alpha, okay, P2 alpha, B2 alpha, I'll explain what does this be, okay, and you multiply it with a 20 possibility. Third stage is a computably search. What we will doing? We will search for sequences that's similar to what we have, okay? All sequence, you know, we had scores, we had 3D structure profile, okay? We had scores for each amino acid, so we can choose which amino acid will be in each position, and then we can get a sequence, 1D sequence of only amino acids placed together. So we can search in the database for sequences that are similar to these amino acids, okay? Which one of them is the most similar one? And that's how we can get from the 3D structure to 1D structure, okay? In so simple talk, okay? That's it for me. That's what I have prepared, okay, about quantum simulation relating to biology. This was a method. Many techniques we can do and we can use, okay, and servers. But the time won't take it all, okay? Just one thing I need to say, and I need to mention, in Spain, 2017, okay, universities there having a lab worked on quantum computers that simulate a life, a real life. What's the meaning of that? You know, each one of us composed of two main things: genotype and something called phenotype. Okay, You have a genome, and this genome can be transcribed and expressed into phenotype, your features and expression of proteins, okay? So they placed all the genome on one qubit and all the phenotypes in another qubit and let them live together, okay, in supercomputer related to IBM. And they let them evolve and mutate, you know, and reproduce and then die. Okay, like playing Sims, if you played it before. Okay. After that, they have taken you know, notes and results, and when compared to real life and experiments, they found it so close. So they are now proceeding in this area, and they are trying to expand the qubits. Okay. So what came on my mind while I'm doing this? Why not we do like, I don't know, quantum computers that contain 20 qubits, okay, for the 20 amino acids, and throw all the amino acids inside it, okay, and let the probability determine what will happen. Okay, this will save time and effort, I think. That's it. Any question? Any question?
Thanks for the interesting talk. Is there questions?
So is this the limit of number of atoms that can be like simulated? Can it go more than 3,000 or like is the optimal number of atoms that can be simulated for this model that you have talking about?
You know, it doesn't have a certain number. Okay, it can work on any number you have. Okay, because you know you have a 3D structure, you don't know what will the final result of it. Okay, it can add more side chains, and so your molecule will grow. Okay, so ITASA and Zappination modeling, it doesn't care what's the number of the atoms. Okay, it only uses 20 amino acids. Okay, because we don't have more than 20 amino acids in the nature. Okay, but it sums them up in side chains and backbone, and then give you 3D structure. If I understood you right, yes. So that
yeah, that answers. But
yes.
So this H1N1 virus, is it what you are simulating in computer by modeling it in terms of amino acid? Okay. If I understand you correctly, is it true?
Yes, we are simulating them. Okay, we are making in silico models of the 3D structure of a protein. Okay, but we cannot use them because we need to validate them at first. You know, the protein data bank was accepting predicted models. That was 10 years ago, I think. But now they are not accepting anything because they need a validation, okay? And they need strong validation. And to me, I can say that nothing is valid. Nothing of this is valid by the nature degree, okay? You need to get the real structure, solve it, and compare it to the model. And if it was so related and so valid, you can go and you can place it in the BDB. That's it. So.
Yeah, so can I ask one? So how long it take time to do this simulation?
From ITASA, you know, I modeled about 52 multiplied by 7. I think 7 to about 400 amino acids residues. Okay, it took about 3 days to simulate them, and so long. If you tried, I tried 28 amino acids, and took about 4 hours. So I think it takes so long. So if we have quantum computers entering this area, I think it will be so fast. So.
I think I don't need the mic.
Okay.
So, when we talked in private about this. I asked you whether you really see any concrete connection to quantum algorithms, because you see, a quantum computer is not a universal machine that accelerates any type of calculation.
Yes.
It has to be dedicated to certain types of algorithms where the quantum advantage really enters the play.
Mm-hmm.
Here, of course, if you are searching databases, like the Brookhaven one with the structures of proteins, the question is whether that can be accelerated by some kind of a Grover search. Do you know whether there's some literature about that?
You know, not a lot, but I read yesterday something about that in Spain University, and they used something related to that, okay, in biological environment, using genomes, which is so related and so close to the work of proteins, okay, using genomes and placing all of them on qubits, and the phenotypes and the features of the people on another qubit, and it worked. So I can expect that we can do the same. I didn't go deeper in it.
Are there any specifics of how they encode genomes in qubits?
No.
This is really, it sounds a little bit like a buzzword if you make it that short. Do you know any detail? Is there any
No details. No, I'm just, you know, I say I'm only a user for now, okay, because I'm a master student, not more. But I can give you the paper, okay, and you can read it, you can understand it, and you can illustrate it. Maybe this will be better. You will have a better understanding more than me, far more better.
So as far as I know, some of the current trends simply are to simulate larger parts of the protein just by classical molecular mechanics and force fields, maybe with a little bit of empirical, semi-empirical quantum theory entering. So that's, I think, also what enters these charm force fields, for which Martin Kaplus got the Nobel Prize a couple of years ago. Mm-hmm. But when it comes to simulating and calculating reactions, which are fast, then people try to really put all the quantum mechanics into the reaction center. And therefore it's, so to say, something like a separation of timescales. So the slow-moving parts, they are simulated classically, with minimum quantum ingredients to keep it computationally doable. And then for the fast part in the reaction center, you really enter the entire quantum mechanics, and you have superpositions, coherences, excited states in a quantum Hilbert space, and so forth and so forth. So that's where the quantum bit enters biology these days. Leave alone, of course, that probably all of you are familiar with Martin Plenio and Patrick Rebentroost's, Musseni's issues on fast processes in the light harvesting complex, where phase noise is beneficial to really gear the exciton into the sink you want to have it in. And
many of the algorithms that I've seen for quantum chemistry, they also come to the hybridization of atomic orbitals in a given molecule. This is the main problem, that we don't kind of calculate efficiently in this. So if I had to guess also where all this quantum computing business will get in, would also be the first principle calculation of a regular molecule, in this case, that they can go with a couple of atoms all the way to the proteins.
Yeah, but of course, in proteins, it doesn't pay to really go ab initio. You have to really divide things into portions where you want to be quantum to really get the essence, and then remainders where it suffices to just take a classical approach. But you don't know about any specifics in this when it comes to the folding problem?
No. No. For now, no.
Are there some things that cannot be calculated right now in terms of them taking too long?
I think it can be everything can be calculated, but it takes a long time. So it's all about fasting the process. That's what I'm thinking. That's it. But it's only about speed, I think, for now. Nothing cannot be done using supercomputers and fast computer workstations, but everything can be enhanced, I think, using quantum computers, I think.
I think that some protein folding is already a problem if you want to do efficiently. This would be something that you cannot, even if you have a supercomputer, it's hard to calculate in an amount of time you're willing to wait.
Right.
There is also interesting, and that brings us a little bit closer to quantum simulation. From the mathematical point of view, there's something in protein folding, in the protein folding problem, which is known under the name Leventhal paradox. And that simply is, if you're trying out all the possible folding motifs, this is far too slow to explain the very fast timescale on which protein folding proceeds. And one of the dogmas in biology, folding molecular biology, is that in principle the structure is totally encoded just in the primary sequence of amino acids. And of course also in the cell, the folding process is catalyzed by ribosomes, or it takes place just at the ribosome.
Yes.
It's being post-processed by chaperones who, the chaperones trying to prevent misfolding and so forth and so forth. And therefore this dogma of molecular biology.
Who will do the job of the chaperone and the... Yes.
Is under fire currently. And it's a little bit similar to these glassy types of phases of matter, which, so to say, also arise because obviously from a time scale, there is no way for nature to really find a global ground state in the amount of time, and therefore it keeps flexible, and we have a glassy phase, and this is very much reminiscent of this type of Leventhal paradox, where also the structure folding thing is too slow from our current understanding.
Yes.
But nevertheless, there we know empirically that proteins have a well-defined structure in which they show up. And therefore, in that case, the Leventhal paradox is indicative of lack of knowledge. And of course, when it comes to really sampling this entire cooperativity of a kinetic cascade of fast processes, slower processes involved in the protein folding, these smart handlings of multi-facet correlation functions, as we heard yesterday from Laura, might be a very inspiring tool in
Yes, I'll try. but I think what you said is the main dogma really in protein folding, and the real issue is that we don't know what will do the job, who will do the job of its chaperones and ribosomes outside the cell. Okay, how will we prevent the misfolding of proteins? How will we see the real time of the protein folding? Okay, is it possible? Even is it possible? Okay, so I think that algorithms can be written to try to describe the process. I think this is the future of this field. Yes, we need to write new algorithms to describe this, finite time processes, and we need to explore more in the proteins and in the incellular life in order to have a better understanding of what's helping the process. Okay, it's surely not only the matter of a chaperone or a ribosome that is getting some RNA related to him and going over it. Okay, mainly, I think there is a missing point that if we got it, we will understand it by a way that we can simulate it, and we can simulate it effectively. That's it. I think that's the future.
Yeah, but it's also intrinsically very, very complicated because
Yes.
it's a kinetic process, this type of folding, and typically the fast components
So
in
fast,
the
yes.
kinetic process are the most difficult ones to spot. And it's always, there are easy tests established in biochemistry that, so to say, spot the time limiting, the slowest part of a kinetic process.
Mm-hmm.
And the fastest ones are very, very difficult
To be spotted, yes.
also from a quantitative point of view. And I think that's also where new ideas are really sought for.
Yes, we need to understand it well, because when I'm thinking in it, everything that we understand well, we can simulate. If we understand something in a really good way, we can simulate it with a mathematical equation. We can use mathematics and the physics to simulate it and to optimize it. Okay.
The problem in these kinetic processes, for instance, is you can simulate that, but it's by far not unique.
Yes.
And you can insert as many transition states as you want, as long as they are, so to say, just reached very fast. And that's exactly the problem in this type of kinetics. So only these alleged transition states of the time-limiting ones,
Mm-hmm.
that's easy to analyze from a mathematical or kinetic point of view. And the fast ones are not unique, and it's very, very difficult to judge upon such a hypothesis whether a certain transition state is part of the folding process or not.
Yes, and that was the reason behind the SAVE servers and all the servers like it. In order to verify anything that we are doing theoretically, okay, we need to validate and to verify if it was right or not. Okay, if it was a powerful tool that's called ITASA and the other ab initio modeling for proteins, if they were so powerful and so correct, I think these servers will be a little lower number, okay, because you won't be going for validation so much, okay. You will only get the protein structure and then try it in the lab, and it will be unique, and you will know the real structure without going through the processing of validation, and after that docking and molecular dynamic simulation and things. That's it.